complications during her pregnancy, but her daughter Evelyn was infected in the womb with a virus that caused deafness and likely brain damage.
Part of what dealing with this virus is that it can happen to anybody. It can happen to a very healthy person like me.
The cytomegalovirus, or CMV, is common in children and adults, but usually harmless. In rare cases, it's devastating for infants with mothers infected during pregnancy.
Now, the challenge we have is because the infection is asymptomatic, you don't know.
Assalamualaikum warahmatullahi wabarakatuh. Hello everyone. After watching our introduction video, I assume most of you or even all of you are getting more and more curious regarding the cytomegalovirus topic. But before we're heading into the material, let me introduce me and also my college. My name is Rifki Fauzi. My colleagues are Muhammad Faisha Arshafi Fahmi and also Raina Sandra Mahira. So, without any further ado, let's go straight forward into the material. I would like to introduce you to the cytomegalovirus, or also known as CMV, which is a very common virus in human, and it is belong to Herpesviridae family. So this virus can be transmitted through various body fluids such as saliva, blood, urine, and also can be transmitted through sexual contact, or maybe it is inherited from the mother to the fetal, or we can also call it as a congenital cytomegalovirus infection. Usually, if this virus infects someone with a good immune condition, there will be no sign of symptoms, or maybe there is only mild symptoms. But if it is infect someone with an immunocompromised condition, it can cause many complications, such as pneumonia, retinitis, and also gastrointestinal disturbance. For the next capture, we're going to talk about the epidemiology of cytomegalovirus infection. In this subcapture, I'll divide into two main points. The first one is the global prevalence of cytomegalovirus infection, and the second one is the prevalence or the epidemiology of congenital cytomegalovirus infection. So, the first one, we're going to talk about the global epidemiology of cytomegalovirus. So, the prevalence of cytomegalovirus infection itself is highly variative. It ranges from 45 until 100%, and it is influenced by geographic location factor and also socioeconomic factor. Besides that, country, usually developing country, tends to have higher prevalence if you compare to the developed country. The second point, we're going to talk about the prevalence of congenital CMV infection. As I already mentioned before, congenital cytomegalovirus infection, it refers to cytomegalovirus infection which is maternally inherited from the mother to the fetal, which is the leading cause of congenital infection worldwide. The global prevalence of congenital CMV is estimated for about 0.67%, with higher rates observed in low and middle income countries compared to high income countries. In Jakarta itself, a study conducted, the prevalence of cytomegalovirus congenital infection is approximately for about 5.8% from the 411 newborn baby. For the next capture, we're going to talk about the morphology of cytomegalovirus itself. So, the first one, we're going to talk about size and shape. The CMV variants are approximately 150 until 200 nanometers in diameter, making them among the largest human herpes viruses. The variant has a roughly spherical shape with an icosahedral capsid as its core. The second one, we're going to talk about the capsid. The inner capsid is about 100 nanometer in diameter and consists of 162 capsomer. This capsid encloses the linear double-stranded DNA genome of the virus. The third one, we're going to talk about the tegumen. Surrounding the capsid is the tegumen, a proteinaceous layer containing viral protein that play roles in viral replication and modulation of the host immune response. The last one, we're going to talk about the envelope. The outermost layer of the CMV virus is a lipid bilayer envelope derived from the host cell membrane. This envelope is embedded with viral glycoproteins essential for host cell recognition and entry. For the next capture, we're going to talk about the culture medium and also the incubation condition. So, the first one, we're going to talk about the culture media. Due to cytomegalovirus is an obligate intercellular virus, its isolation process requires living host cells for replication. For example, we can use fibroblast cells, which is commonly used as host cell because they are sensitive to CMV infection and exhibit characteristic cytopathic effects. To survive and proliferate optimally outside the body, these fibroblasts require a nutrient-rich culture medium. We can use the MEM and also MEM, which stands for Dulbecose Modified Equal Medium and Minimum Essential Medium. Both of them serves as a nutritional environment that supports the survival and growth of fibroblasts, thereby enabling CMV to infect and replicate. And then the second part, we're going to talk about the incubation condition. The first one, we're going to talk about the temperature. It is 37 degrees Celsius to mimic human physiological condition. And the second one is atmosphere, 5% carbon dioxide in a humidified incubator to maintain pH balance in the culture medium. And the last one, the duration. CMV has prolonged replication cycle. Cytopathic effects or CPE typically become evident between 7 until 14 days post-infection, depending on the viral strain and multiplicity of infection or MOI.
Okay, now the next thing that we're going to discuss is the characteristics of the cytomegalovirus culture growth. So there are three things that we're going to discuss. The first one is the cell type that is used, the second one is the growth rate, and the third one is the cytopathic effects. So the first one, the cell type that is used is the human fibroblast cells. Why? Because the human fibroblast cells are more susceptible towards CMV than any other virus. The cell type that is used is the human embryonic lung, the MRC5 or the WI38 fibroblasts. The second thing that we're going to highlight is the growth rate. So the growth rate, the cytomegalovirus is a slow-growing virus. The cytopathic effects, which are the social characteristics, may take about 1 to 3 weeks to be visible. And the detection technique that is used in the cytomegalovirus culture growth is the shell vial assay. And detection may take about 24 to 72 hours. The last thing that we're going to highlight is the cytopathic effects. So the characteristics of cytopathic effects contains of cytomegaly, which is the enlarged cell. And the second one is the owl's eye, or the intranuclear inclusions. These are basophilic and also dense. And the third one is the cell lysis and destruction of the cell monolayer over time. Another thing that I'm going to highlight is, as you can see in the picture, there are arrowheads. So the arrowheads show the nuclear body inclusions, which is also one of the hallmarks of cytopathic effects. Okay, now let's discuss about the antigen structure of the cytomegalovirus. So the antigen structure of the cytomegalovirus contains of 4 components. The first one is glycoprotein B. Then there's the second one, which is also a type of glycoprotein, which is glycoprotein H or L. And then there's the third one, which is a type of tegument protein called PP65. And the last one is the immediate early protein 1 and immediate early protein 2. Now let's discuss from the glycoprotein B. So the glycoprotein B is essential for viral entry into host cells and are a major target of neutralizing antibodies in our immune system. Now let's move on to the second type of glycoprotein, which is glycoprotein H or L complexes, which facilitate membrane fusion during viral entry. And then we're going to move on to the tegument protein, which is the PP65. So the PP65 is a tegument protein and a dominant target for cellular immune responses, especially cytotoxic T cells. And now let's move on to the last antigen structure, which is the early immediate protein 1 and 2, which regulate viral gene expression and are early markers of infection. These antigens are crucial for CMV diagnosis, vaccine development, and also immune monitoring in clinical settings. Okay, the next thing that we're going to discuss is the overview of diseases caused by the cytomegalovirus. So it is divided into 3 subsections. Diseases caused in healthy individuals, diseases caused in immunocompromised individuals, and diseases caused in congenital individuals. So in healthy individuals, the cytomegalovirus typically causes fatigue, fever, sore throat, and swollen glands. In immunocompromised individuals, the cytomegalovirus might cause the rhinitis, which is the inflammation of the kidney, the colitis, which is the inflammation of the colon, pneumonia, which is a lung disease, and hepatitis. In congenital individuals, cytomegalovirus might cause microcephaly, seizures, vision impairment, and hearing loss. Okay, so now we're going to explain about the pathogenesis of the cytomegalovirus based on the picture that I've shown. So the process begins when CMV binds to the surface receptors on the cells, activating integrin, which then triggers a cascade of intercellular events. So as integrins activate, they lead to the induction of cellular morphological changes. The shape and structure of the cell alter and become an environment more beneficial for the virus to replicate. So these morphological changes further lead to the activation of signal transduction pathways, allowing CMV to manipulate cellular processes for its own replication and survival. Importantly, after the initial infection, CMV has the ability to remain latent in mononuclear cells such as B cells, T cells, and macrophage. This is the key to CMV's persistence in the human body. It can stay hidden for years without any symptoms showing. However, under certain conditions, especially in immunocompromised individuals, the virus can reactivate. This reactivation leads to cell damage as the immune system responds and infected cells are destroyed. Finally, damage and immune response produce clinical manifestations that vary depending on the affected organ, such as rhinitis, colitis, and pneumonia in severe cases.
Diagnosing CMV infection depends on the individual's health status and the presence of the symptoms. For the first one, there is laboratory test. Laboratory test is taking body fluids such as blood, urine, saliva, or other body fluids, as well as the tissue sample to see CMV virus itself or antibodies against it in the body fluid. There are a few types of laboratory tests. For the first one is serological test, which it detects the IgG and the IgM antibodies. It detects CMV antibodies in the blood. A positive IgG indicates that this patient already has a past infection, while IgM may suggest a recent infection or reactivation. The second one is PCR. PCR is the fastest laboratory test. It detects the CMV virus's genetic material, or DNA, in various bodily fluids or tissues. PCR on saliva is the standard for diagnosing congenital CMV infection in newborns. The next one is antigen detection tests, such as the PP65 antigenemia assay can detect active CMV infection by identifying viral proteins in the blood. The last one is histopathology. Examination of tissues biopsies can reveal characteristic cytomegalic inclusion bodies indicating CMV infection. So actually there are special conditions for diagnosing CMV viruses. For the first one, when we diagnosing a newborn, which congenital CMV, testing within the first 3 weeks of life is crucial, with the gold standard test, which is PCR on saliva and urine, to primarily detect the CMV virus. The second one is diagnosis during pregnancy. Testing pregnant women for CMV antibodies can determine if they have had a past infection. A new infection during pregnancy may warrant amniocentesis to test the amniotic fluid for fetal CMV infection, especially if ultrasound findings suggest possible CMV-related abnormalities. And the third one is diagnosis in immunocompromised patient. Regular monitoring for CMV may be necessary. Detection of CMV in blood or other body fluids along the clinical symptoms helps diagnose active infection. The overview of therapies or treatment for this infection is actually no needed for the healthy individuals since it shows no sign and symptoms. But if it happens to newborn with symptomatic congenital CMV, first, the antiviral medication like ganciclovir injected intravenous and its oral prodrug valganciclovir are the drugs of choice. Treatment typically lasts for 6 weeks to 6 months and may help improve hearing and developmental outcomes. Regular monitoring for side effects like neutropenia is also essential. For the immunocompromised individual, treatment is often necessary to manage active infection and prevent severe complication. We can see here for the first line antiviral agent, for the first one is ganciclovir, which is injected by intravenous, and it is the effective antiviral for CMV. The first line antiviral agent is valganciclovir, an oral product of ganciclovir commonly used for maintenance therapy due to its improved bioavailability. And there is also alternative antiviral such as foscarnet, it used in cases of ganciclovir resistance or intolerance. And the second one is cidofovir, another alternative for resistant CMV strains, and the third one is latermovir, approved for prophylaxis CMV seropositive allogeneic stem cell transplant recipients. It inhibits the CMV terminase complex. How to prevent CMV infection? For the first one, of course, keep your hand clean by doing the frequent hand washing. Wash hand thoroughly with soap and water for 15 until 20 seconds. Second one, avoid contact with saliva and tears. When interacting with children, avoiding kissing them on the lips. Kissing on the forehead is a safer alternative, especially for pregnant women. Not share items. Avoid sharing food, drinks, utensils, and personal items like toothbrush, especially with young children. Next one, clean surface and toys. Regularly sanitize the toys, countertops, and other surfaces that may come into contact with children's urine or saliva using simple detergent and water. water. Practice safe sex. Use condom. Be cautious with body fluids. When disposing of diapers, tissues, and other items contaminated with body fluid, wash hands roughly afterwards. For pregnant women, maintaining good hygiene is particularly important to reduce the risk of primarily CMV infection during pregnancy, which carries the highest risk of congenital CMV. Blood and organ transfusion. Screening of blood and organ donors for CMV can help prevent transmission in transplant and transfusion settings. Last one is prophylactic antiviral in certain high groups such as CMV seronegative transplant recipient of organ from CMV seropositive donors. Prophylactic antiviral medication, for example, valganciclovir, latermovir, are often used to prevent CMV disease.